Okay, the first topic that I'm going to talk about is called homeostasis. So this is your body's ability to use feedback loops in order to maintain a certain way of living. And so it's feedback loops. So there's two kinds of feedback loops. There's negative and then there's positive. So negative feedback loops are described as negative not because of its like moral value, but it's described as just moving opposite or against whatever stimuli your body is experiencing. And so as opposed to negative, the opposite would be positive. So this is moving with the stimuli that's being presented. And so as an example, if the temperature outside were to move up, then your body's temperature is going to decrease. And so they're obviously moving against each other. The results are not the same as a stimulus, so this would be a negative feedback loop. Positive is not quite as simple. It has two different possible ways of being positive. There's intentional and then there's unintentional. Intentional happens only about 5% of the time. One really good example of this is when someone is going into labor, they are like the stimuli that they're receiving is that the baby's head is pushing on the cervix, which sends a signal to your brain, obviously. And if it were a negative feedback loop, they would work opposite to the stimuli, so it would try to prevent that from happening. But because this is a positive feedback loop and we're working with the stimulus, then that cervix is going to relax and then you're able to deliver a baby. However, that does only happen 5% of the time. The other 95% of the time, it is unintentional. So this would be like if the temperature outside were moving up, then your body's temperature would also be moving up, which is obviously something we don't want to happen. So if we were to draw like a visual of this, we have a stimuli, and this is picked up by different receptors, whether they be pain or temperature or your eyes or anything. Like these are just receptors in the body, and then the receptor sends signals to the control center, and then the control center sends signals to the effectors. So the signals that the receptors send to the control center are called your afferent pathways. And then the control center, the signals they send out to the effectors are called efferent pathways. So when we're talking about positive feedback, this is also referred to as broken effectors. So this part is not working for them because this guy sends signals back to the receptors and tells the body how to handle the situation. And so when it's negative, it's like, yo, do the opposite. And then if it's positive, it's obviously telling it to do exactly the same. So most of the time we don't want this to happen because this is not helping us maintain our way of living. We can also draw a chart to help better explain this. If I made a chart and this is body temperature, I'm just going to abbreviate for time here. And then this is time. And then every one of these notches is an hour. If we just took the temperature of someone every hour, it wouldn't be exactly the same all the time. It would fluctuate, like so. But it stays within this normal-ish range. And so this is where we want to be. This like average line, this is what we refer to as our set points. So homeostasis is trying to maintain these set points even when we're receiving different types of stimuli and are in different environments. The next thing I wanted to talk about is kinetic energy. So every cell, every molecule, everything in the whole universe has something called kinetic energy. And this is just its ability to move, basically. And so when we're talking about things moving across the bilayer of the cell, we need to talk about kinetic energy and how it's using that energy to move. So there's a couple questions we can ask ourselves to decide if something, like what kind of diffusion or movement something is doing. And so our first question we can ask is that all? Is all the cell using Is it just kinetic energy, or is it adding something else? So if that answer is yes, and it's only using that thing, then this is called, there's two different ways of this. This is called simple diffusion. This is solely just against gradients and just because it naturally wants to move that way. However, there is another type that needs a little bit more of facilitation, so that one's called facilitated diffusion. So the differences in between simple and facilitated diffusion is they're both small molecules, but these are not charged, and these ones are charged. So facilitated diffusion has protein mediators, and the most common of these are going to be mechanical channels, voltage channels, and then ligated channels. So all three of these things need different environments in order to open up or close, and this helps things move with their gradient. So that's going to be the biggest difference between kinetic, like simple facilitated diffusion, versus primary and secondary diffusion. And so to answer the first question again, if that's no, then there are going to be two different types of this. And to differentiate between the two, the next questions we can ask ourselves is, are we using the energy directly or indirectly? And so the energy we're talking about is ATP here. And so if we're using it directly, we're talking about primary diffusion. And this, I'm going to draw a really simple picture here. This is going to be our sodium-potassium pump. Because this pump needs to be using ATP, and the ATP is directly in contact with this pump, and that's what it's working on. However, there is another type of diffusion that uses energy, but it's a lot more indirectly. So this is going to be called secondary diffusion. This is another type of pump that is found on the cell, and this is going to be moving calcium in and other types of molecules, but it's using energy that this is also using. So the ATP isn't exactly working with this pump, but it is still using the ATP and using that energy. So that would be classified as secondary diffusion. So those are the four different types of diffusion that move things across the cell layer. Next, we have something called osmolarity. So this is super important when we're talking about how things move across cells, because it describes kind of what's happening. However, there are different ways to talk about particles and stuff. And so the main difference between osmolarity and molarity is, I'll just make a quick note over here, that molarity is just talking about concentration, or like how many moles there are of a substance, whereas osmolarity is talking about the amount of particles in a certain type of substance. This is going to be really important because this will affect the substance that it's going into, especially when we're talking about putting things into or out of blood. And we also use a word called tonicity when describing the diffusion of water and particles, especially around cells. The differences between osmolarity and tonicity is that osmolarity is, again, just about particles and how they act, but tonicity is talking about the movement of water, and specifically how the osmolarity is going to affect this moving water. And so with these two definitions, we can have something that's isoosmolar. So it has the same osmolarity of whatever it's going into, because this term is used in reference to two separate things. So their osmolarity is going to be the same. However, it can also be hypotonic at the same time. In this case, I'm talking about something called 5% dextrose. It's a type of sugar. So in the beginning, this is isomosmolar because it has the same osmolarity as blood, which is usually how we give 5% dextrose. However, because it is permeable to the cell membrane, this is going to enter into the cell and get digested. And then this is going to create a hypotonic area out here in the extracellular fluid, which is going to cause the water to move, as opposed to saline, which isn't permeable to the cell. So that would be isoosmolar and isotonic. And then when I talk about iso, I mean the same. And then when I say hypo, I mean less. So that is the differences between osmolarity, tonicity, and molarity, and how it is used in references to different types of substances and what we're using them for.